Hello everyone, I'm welcoming you everyone to this amazing platform of PW NEET English, and you know that we have started a quick revision series of physics, chemistry as well as biology. Hope everybody, all of you are doing absolutely great in your life and preparing for upcoming NEET exam. And in today's class, we'll be covering this particular chapter, that is biotechnology. It's a unit actually. There are two chapters: biotechnology principles and processes, and biotechnology and its applications. Yes. So we'll be discussing few of the important PYQs, as per the trend analysis we have chosen few important questions, and for today's lecture we'll be completing 50 most important questions from these two chapters. And after completion of today's session, I'll be giving another 50 questions which will be available in our WhatsApp group. So if you are not inside our WhatsApp group, please search the link you will be finding somewhere inside our YouTube channel, that is PW NEET English. Go get inside, join our WhatsApp channel, and after every lecture we are uploading the handwritten notes as well as 50 important questions for your homework purpose. Yes, very good evening, Dr. Grishma. Hello, Priya Kumari. How you are doing? So let me know in the comment section, and hope preparation is at the peak and heart is really beating fast because NEET 2026 is coming really closer to us. So if you are ready, I'll be starting today's session. So Dr. Grishma, is it your real name? And you are watching from which part of the country? Okay. Yes, Ayush, very good evening, very good evening. Welcome back to the class. So let's start today's session. So first question, we'll be solving that the TI plasmid of Agrobacterium tumefaciens stands for tumor inducing plasmid, temperature independent plasmid, tumor inhibiting plasmid, or tumor independent plasmid. So you can give your answer. Just write down in the comment section as A, B, C, or D. Now this poll will pick your answer from the comment box itself, and it will be displaying the leaderboard. Okay, so you are from Telangana. Great. So pastel palette is given the answer, that is tumor inducing plasmid. Yes, exactly, TI for tumor inducing plasmid. Let's see anyone of, so only we got one answer, and all of you, most of you have given this particular answer only. So tumor inducing plasmid is having a particular piece of gene called as that is called as tDNA, and because of the expression of the tDNA, plant cells will be getting will becoming cancerous in nature. Now what you have to do, we have to manipulate that tDNA from the TI plasmid and will be inserting gene of interest, and will be utilizing Agrobacterium tumefaciens as a natural genetic engineer as a vector. Next question, which of the following statements is incorrect? Bioreactors are used to produce small scale bacterial culture. Bioreactors have an agitator system, an oxygen delivery system, and foam control system. A bioreactor provides optimal growth conditions for achieving the desired product. Most commonly used bioreactors are string type, a steroid type of bioreactor. So here I'm starting the poll here. You can answer accordingly. Okay, Ayush Pastel has given the answer, that is option A. So according to you, bioreactors are used to produce small scale bacterial culture. That is incorrect statement. Yes, exactly. that is the answer. So generally we are using bioreactors for large scale production of our products. It can be any sort of enzyme or protein or antibodies or something else. Okay, coming to the next question, question number 3. The gene gun method used to introduce alien DNA into host cell, microparticles of dash metals are used. Another easy one. Here I am sharing the poll, so you can write down your answer in the comment section. So we are talking about gene gun method. We are bombarding DNA and RNA along with metallic particles directly inside the cytoplasm of plant cells. Okay, so here I can see the answers given in the comment box. That is D. Yes, Ayush, Pastel, Christina and Dr. Grishma. Okay, all of you have given right answer, and that is absolutely perfect one. Tungsten or gold coated particles of DNA and RNA are bombarded, penetrated with the help of the gene gun or ballistic gene gun directly inside the plant cells. Okay. Now let's move forward to the next question, and the next question is here: ligation of foreign DNA at which of the following site will result in loss of tetracycline resistance of PBr322. Here they are talking about plasmid of Bolivar and Rodriguez, and here a situation is created like that. You have to perform insertional inactivation by inserting gene of interest into the tetracycline somewhere inside the tetracycline resistant genes, means TET-R gene. So can you answer this particular question? Okay, again all of you are giving absolutely right answer, that is option D, BAM-H1. Yes, so if we look for this PBr322 plasmid, it has some important sites. Called one site that is called AMPR, ampicillin resistant site, another site that is TETR, tetracycline resistant site. We have ORI site, origin of replication, and we have ROP, repressor of primer site. So this particular site is for AMPR, ampicillin resistant site, and this particular site is for tetracycline resistant, means the product of TET-R gene will be inhibiting the action of tetracycline antibody, and that's containing two restriction site for two different restriction endonuclease enzyme. One is BAM-H1 and one is SAL1. One is BAM-H1 and one is SAL1. So according to our question, if you are performing insertional inactivation, particularly in BAM-H1 site, the bacteria, the recombinant will be, or the transformed bacteria will be losing tetracycline resistance. Let's move forward to the next question, and the question is here, and the question is saying the enzyme A is needed for isolating genetic material from plant cell, and enzyme B is isolating genetic material for fungus. Another easy one from the chapter Biotechnology Principles and Processes. So you can answer. You can write your answer into the comment box. Okay. So well, we are talking about the process of construction of recombinant DNA. Then the very first step, where we need to isolate the DNA from a particular cell. For different cells, we are utilizing different enzymes to digest the envelope. Okay, so I'm getting the answer from Ayush, Pastel, Dr. Grishma and Christina, and the answer is, according to you, that is option C, and yes, that is our answer, option C. So cellulase is used to break down the cell wall of plant cell, and chitinase is used to break down the cell wall of fungi. Okay. Let's move forward. Question number 6. What is the fate of a piece of DNA carrying only gene of interest which is transferred into an alien organism? Okay, so you have inserted a particular gene of interest into another host organism. Now we have some statements following that. The piece of DNA would be able to multiply itself independently in the progeny of cells of the organism. It may get integrated into the genome of the recipient. It may multiply and be inherited along with the host DNA. The alien piece of DNA is not an integral part of chromosome. It shows ability to replicate. Here I am sharing the poll and you can write down your answer. Read it carefully. And another, it's a kind of little bit tricky, moderate kind of question, not easy, not tough, a moderate kind of question. So there is a topic you'll be finding in your NCERT book, and the line is written, the fate of the gene of interest inside the alien organism. So here you have to determine the fate. Okay, so first answer I got from pastel palette, that is as option A. Okay, Raj is giving the answer as option C. So let's check the options one by one. Okay, so the piece of DNA, so we have to find out, uh, correct the correct answer. We have to find out the right ones. Okay, the piece of DNA would be able to multiply itself independently in the progeny cells of the organism. It may get integrated into the genome of the recipient. It may multiply and be inherited along with the host DNA. The alien piece of DNA is not an integral part of chromosome. Okay, it shows ability to replicate, doesn't have the ability to replicate. The alien piece of DNA is an integral part of chromosome. Okay, if you want to be replicated, then you need to make it an integral part of the chromosome. The piece of DNA would be able to multiply itself independently in the progeny cell of the organism. It cannot multiply itself because it doesn't have the oocyte of replication. It may get integrated into the genome of the recipient. Yes, it may get integrated into the genome of the recipient. It may multiply and be inherited along with host DNA. Okay, so here we got the answer option B and C, and the answer is, correct answer is option D. So none of you have given the right answer for this particular question. Okay, so again I am explaining, the independent piece of DNA would not be able to multiply itself because it doesn't have the oocyte. So that's why statement 1 is wrong. Statement 2 is correct because it may get integrated into the genome of the recipient. If it's getting directly integrated into the genome of the DNA of host, then that possibility is there. And if it's getting integrated, it can multiply and inherited along with the host DNA. It's not present as a plasmid. You need to remember this. That's why B and C are correct. The alien piece of DNA is not an integral part of chromosome. Obviously, that alien piece of DNA is not integral part of the host DNA. It's a separate one. It's an alien one, gene of interest. It doesn't have the ability to replicate by its own. Next question. Another statement based question, and the question is, statement 1 is saying, if any protein encoding gene is expressed in a heterologous host, it is called a recombinant protein. Statement 2 is saying, in a continuous culture system, the fresh medium is drained out from one side while the used medium is added from the other side. Let's see who can give the answer. So if any protein encoding gene is expressed into heterologous host, means the alien organism. The host organism is called as heterologous, that's because the genetic makeup of the host organism is not similar to the gene of interest. That's why we are calling it heterologous host. And you can call it as recombinant protein. When the gene of interest will be expressed, we can call that recombinant protein. So it's a true statement. Statement 2 is saying, in a continuous culture system, the fresh medium is drained out. So it's not the fresh medium which is drained out, which is the used medium that is drained out, and fresh medium that is added. So here it will be, answer will be like this: statement 1 is true and statement 2 is false. Statement 1 is correct, but statement 2 is incorrect. So B will be our correct option, and all of you have given the right answer. Yes. Now next question, given below are two statements. Yes, B is correct. So the statements are, first statement is bright red colored bands of DNA in a bacterium chloride stained gel exposed to visible light. The separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. Okay, I'm sharing the poll. I think this is kind of easy or one. You can easily give the answer. Okay. So bright red colored bands of DNA in a barium chloride stained gel exposed to visible light. So yeah, this is question and practice series only. But Praveen, if you have any doubt from any of the topics from these two chapters, you can ask your doubt into the comment section. I'll be explaining that also. Okay, Praveen? Okay. So here I am getting the answer as option B, and you people are saying the statement 1 is incorrect and statement 2 is correct. Yes, exactly. Option B is right answer, and most of you have given the answer perfectly. So why statement 1 is false? As because bright red colored bands of DNA are not observed. First of all, we treat the DNA under ultraviolet ray with ETBr, ethidium bromide, and they appear bright orange in color. So that's why statement 1 is false. And the process of separation the bands of DNA and the cutting out from the agarose gel, that is called the process of elution, right? So statement 2 is true. So that's why option B is the correct one. Now coming to the next question, question number 9. Let's check the question. Given below are two statements. The statement 1 is saying, in the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. Okay. Now what statement 2 is saying, primers are small chemically synthesized oligonucleoside. Okay, look it, check it carefully and give the answer. So if you can remember the lines of NCERT or the concept, it's very easier for you. Okay. So in the year of 1963, two enzymes were isolated from E. coli bacteria. One is called as methyl transferase, another is called as restriction endonuclease, right? Okay. And both of them are responsible for restricting the growth of bacteriophage. One will be cutting the DNA, that is restriction endonuclease, and another, that methyl transferase, will be methylating the own DNA to save the bacterial DNA from the attack of the viral DNA. So I am getting the answer as option B. So people are saying that statement 1 is incorrect. Okay. So in the year 1963, the two enzymes responsible for restricting the growth of bacteriophage of E. coli were isolated. That is true fact. As I said, One is methyl transferase, another is restriction endonuclease, and both of them are responsible for restricting the growth of bacteriophage virus for bacteria. Okay, and the statement 2 is saying primers are small chemically synthesized oligonucleosides. Never. So this is false statement. Primers are small chemically synthesized oligonucleotides. So statement 1 is correct and statement 2 is incorrect. So our answer is option A. Let's check how many of you have given right answer. Okay, so all of you have given the wrong answer for this particular question. So the answer is correct answer is option A. Again, repeating the question. Check it carefully. In the year of 1963, two enzymes responsible for the restricting the growth of bacteriophage. One is methyl transferase, another is restriction endonuclease. They were isolated from E. coli bacteria. So that's why statement 1 is true and statement 2 is false. Even someone is answering as option C. So C is saying both statement 1 and 2 are correct. No, statement 2 is not correct. Why? My dear students, because these primers are oligonucleotides, right? And it's written here primers as oligonucleoside. So oligonucleoside plus phosphate group, that will make the nucleotide, the building blocks of DNA or RNA, right? That's why statement 2 is wrong. Got it? Now we are moving forward to the next question, and the question is saying given below are two statements. Okay, so assertion and reason kind this is. So first check what assertion is telling us. In a sparged stride tank bioreactor, sterile air bubbles are sparged into the bioreactor. The bubbles inside the sparged tank reactor dramatically decrease the oxygen transfer area. So where you are operating that oxygen bubbles, providing the oxygen bubbles inside the liquid medium, so that will be creating the sparging effect. So will that be decreasing the oxygen transfer area or increasing the oxygen transfer area? So if you know the fact, the answer is the cup of tea for you. So let me check how many of you are giving the right answer for this particular question. Already I have got two answers from Raj and Ayush. Okay, so both of them answered option A, and also Ritika also given the right answer. Assertion is true. Sparge tank bioreactor, sterile air bubbles are sparged into the bioreactor. That is absolute true fact. But this bubbling will be increasing the oxygen transfer area, not decreasing. So this is a wrong statement. So that's why we got option A as right answer. Okay, so 100% all of you have given correct answer for this particular question. Now let's move forward to the question number 11, and the question is saying another assertion and reason. ROP sequence, remember the rope gene of PBR322 is present in the PBR322, and reason is saying ROP codes for the lipids involved in the replication of plasmid. Okay, again a conceptual question. If you know the concept, the answer is easy for you. It's about repressure of primer gene, ROP, repressure of primer, or some people call it repressure of polymerase. So again I am getting the answer from Dr. Grishma, from Raj, from Surya, from Ayush as option A. Means assertion is true, rope sequence present in PBR, that's obviously 100% true fact. But ROP, repressor of primer, doesn't code for lipid, rather it will be coding particular protein which will be blocking the activity of the primer, which is responsible for initiating the replication of that particular DNA setup. So that's why reason is false. So option A is correct, and all of you have given right answer. Next question, another assertion and reason you can check here, and the assertion is saying the recombinant DNA molecule cannot be created unless one cut uh, the vector and source DNA with the same restriction enzyme. Okay, and the reason is saying when cut by same enzyme, both will have same ends that can be joined by DNA ligase. Okay, now this is a comparatively easier question. Okay, yes, Ayush, that was for the previous one. It should be proteins instead of lipids. Exactly. Now please try this particular question. Okay. Raj is answering as option B. Ayush is answering as option C. So let me check. The recombinant DNA molecules cannot be created unless, okay, so you'll be needing the similar restriction in enzyme if you are using EcoR1. If you have to cut by utilizing that EcoR1 for your gene of interest as well as the plasmid, wherever you want to insert that. So that's a true assertion. Why? We cut by same enzyme and both will be forming sticky end. So complementary sticky end will be formed, or that will be making the activity of ligase easier to join the different pieces of DNA molecules. So option A will be the correct one. Okay, and I don't know why none of you have given the right answer. Okay, all of you have answered C, both option assertion and okay option. Sorry. Yes, so both assertion and reason are true, and R is the correct explanation of A. So this option is given in option C. Yes, you people are correct. Now coming to the next question, another assertion and reason. Bacterial cells can be made competent by monovalent cations. It increases the efficiency with which DNA enters the bacterium through the pores of cell wall. Now let's see how many of you can give the right answer for this particular question. Bacterial cells can be made competent. What do you mean by competency? Bacteria will be showing reluctancy when you are trying to insert gene of interest inside it. So that's why forcefully you need to tell bacteria, hello bacteria, you have to intake the piece of DNA because I want to make you transformed and I want to make the successful application of our DNA technology. Okay, so bacterial cells can be made competent by monovalent cations or divalent cations. Yes, we have studied, we are using cold medium with calcium Ca++. We need to use divalent cations, not monovalent cations. So assertion is a false fact, wrong fact, incorrect fact, and as because assertion is false for this particular question is referring it increases the efficiency. That means monovalent cation cannot increases the efficiency. So reason will also be false here. So both assertion and reason are false for this particular question. Okay, so 25% of you have given the right answer. I think many of you have overlooked, right, this monovalent cation part. Now let's move forward to the next question. Question number 14, and the question again another assertion and reason. The assertion is saying the presence of an insert into the coding sequence of the beta galactosidase gene, that means LAGZ, results in its inactivation. The reason is saying in the presence of a chromogenic substrate, recombinant colonies produce a blue color. Okay, if you can remember the vector part, the topic called vector And the insertional inactivation concept, this is the easier one for you. Okay. So how many of you actually knows the answer? Let me check. How many of you are knowing the answer? Comment just into the box given below. Okay. So I'm getting answer as option, few are answering as option A, few are answering as option D. So let me explain it. The presence of an insert into the coding sequence, that means the lag Z, right? It is responsible for the production of beta galactosidase enzyme that will be resulting in its inactivation. We call it insertional inactivation. So that's a true fact. But the reason is saying, in the presence of chromogenic substrate that is called as X-gal, the chromogenic substrate, recombinant colonies cannot produce blue color. Rather, normal colonies can produce blue color. Non-transformed bacteria can produce blue color upon reacting with that chromogenic substrate, while the lag Z gene will be producing beta galactosidase enzyme, right? But as because the beta galactosidase gene is inactivated by insertion of gene of interest, it cannot produce blue color. So reason will be false or incorrect here. So here for this question, assertion is true, reason is false. Okay, let me check how many of you given correct answer for this particular question. 50%, almost half of you have given right answer. Next question, another assertion and reason. And what assertion is saying us, DNA cannot pass through the cell membrane. Okay, reason, DNA is hydrophobic molecule. Now this is a easier one. I hope all of you can answer correctly for this particular question. Okay, Ayush, Ritika, DNA cannot pass through the cell membrane. Cell membrane is hydrophobic in nature. Okay, after completing this question, I'm coming to the previous question, Ayush, right? Okay, so DNA cannot pass through the cell membrane. That is exactly true fact in normal condition, obviously. Reason, DNA is hydrophobic in nature. DNA is hydrophobic in DNA is a hydrophobic molecule. Okay, no, DNA is hydrophilic molecule. Cell membrane is hydrophobic molecule. DNA is negatively charged. Cell membrane is also negatively charged. So that's why they will be repelling each other. So DNA should be here. DNA is not hydrophobic. DNA is hydrophilic due to the presence of the phosphate group inside it. So here assertion is true, but reason is false. Yes, option A. Let me check how many of you have given right answer. So 100% of you have given right answer. Now I'm going back to the previous question. Okay, Ayush is having some doubt for this particular one. So see, Ayush, assertion is saying the presence of an insert in the coding sequence of the beta galactosidase gene result in its inactivation. Let me clarify. So if this is the bacteria, and inside this bacteria you kept a particular plasmid which has a special gene called lag-Z. Remember, the gene is called lag-Z, right? Now this lag-Z gene, the product of lag-Z gene will be beta galactosidase enzyme. The product of lag-Z will be beta galactosidase enzyme. Now you know that every enzyme is having its own substrate and it will bind with each other following the lock and key kind of or induced by following that induced feed model theory, right? So in the this is a non-transformed. This is the original bacteria, original E. coli you have in your lab just before performing the rDNA technology. Now, if as a chromogenic substrate you are putting X-gal, a special chromogenic substrate, that means that is having the capability to produce color when it will interact with this beta-galactosidase enzyme. So, when this X-gal will interact with beta-galactosidase enzyme, the medium will produce blue color. The medium, inside the medium you can observe production of blue color, means blue color is denoting presence of intact laxate gene or beta galactosidase gene. Remember this. Now what you have done? Now what you have done? You have inserted the gene of interest. You have inserted the gene of interest inside the laxate. So what will happen now? The laxate gene will be losing its capability. Laxate gene will be inactivated soon, and as soon as laxate is inactivated, it cannot produce beta-galactoside enzyme, right? If beta-galactoside enzyme is not produced by this bacteria because laxate gene is broken, inactivated, because you have inserted another gene inside it. So if you keep on providing more and more chromogenic substrate or X-gal, then there is nobody to react with this. So if X-gal is not reacting with beta-galactosidase, it will not be producing blue color. So that is the identification of transformed bacteria. That is the identification of transformed bacteria. Your rDNA technology is successfully helped you to make a bacteria to transform it. So non-production of blue color is indication of transformation. Production of blue color is indication of non-transformation, and that's why I said, as per this question, so reason is saying that in the presence of chromogenic substance, recombinant colonies produces a blue color. So that's why it's a false fact. In the presence of chromogenic substance, recombinant colonies cannot produce blue color. Rather, the native colonies can produce blue color. I think now you got my point, Ayush. Now moving forward to the next question. Someone is the name I cannot read, W4J7H, and before that, yes, hello, good evening. Moving forward to the next question. Okay, this question number 15 is also done. Now question number 16. Another assertion and reason. Okay, so tDNA, another question from tDNA. tDNA transforms normal plant cell into a tumor. So part of Ti plasmid, tumor inducing plasmid. Reason is saying retroviruses in animals have the ability to transform normal cells into cancerous cells. Okay, so two different kinds of statements are there. Let me check, either you can answer this properly, then I'll be explaining this. Santosh, sir, when will classes start on YouTube which will have concepts in English? So, Santosh, if you are new to this channel, that is PW NEET English, I don't know in which class you are studying right now. We are coming with batches. Arjuna for class 11, Lakshya for class 12, means Arjuna English, Lakshya English, and Yakin English for long term or dropout students. And Lakshya and Arjuna batches will be starting from this coming 13th April. So you can check into our PW app and you can get enrolled. Or if you are preparing for this year and if you want to revise back all the chapters, you will be finding, you have to check the playlist section of PW NEET English, and every chapters of physics, chemistry, botany and zoology are given in one short and crisp manner. If you only study those chapters, I think your 90% preparation will be over. Okay. So I'm getting an answer. A few of Ritika is answering as option A, Ayush answered as option D, Dr. Grishma D. Okay. So tDNA transform normal plant cell into a tumor. That is true fact, because tDNA is, that is a part of tumor inducing plasmid, a part of Agrobacterium tumefaciens, the natural genetic engineer. Reason retroviruses in animal have the ability to transform normal cells into cancerous cell. That's also true. Due to the influence of V oncogene or viral oncogene, few of the retroviruses, they have this capability. But these assertion and reason, they are not related to each other. So that's why, yes, both A and R are true, but reason is not correct explanation of A. That is the right answer. and none of you, okay, few of you have given the right answer. Okay, in the comment section. Now let's move forward to the next question, question number 17. And another assertion and reason: vectors should have preferably single recognition sites, direct line of NCERT, the commonly used restriction enzymes. Okay, reason is saying presence of more than one recognition sites within the vector will generate several fragments which will make the gene cloning easier. Sharing the poll here, you can write your answer. Rithika. Okay. Now for the previous question you were asking that DISM retrovirus that is used for transforming animal cells, right? But normal virulent retroviruses, they have the capability to cause cancer. That was written in the reason. Okay, now look for answer of this question. Okay, Ayush is saying the answer should be option A. Okay, so vectors should have preferably single recognition site, absolutely true, for a particular restriction enzyme, and presence of more than one recognition site within the vector will generate several fragments, and that will create a nuisance for you, that will make your job much harder, so that will not make your job easier. So reason will be wrong or false here. So assertion is true and reason is false. Option A, we got our right answer, and 100% of you are correct in terms of your answer. Let's move forward with another assertion and reason kind of question. Okay, uh, assertion is saying Escherichia coli having PBR322 with DNA insert at the PstI site, if you can remember the diagram of PBR322, cannot grow in a tetracycline medium. Okay, and the reason is saying the recognition site for PstI is present in the ampicillin region of PBR322. If you can recall the diagram, the question, the answer is cup of tea for you for this particular question. Okay, so already I got the answer from Ayush. According to Ayush, it should be B. Okay, let me check more answers from you people. Anybody else who want to answer for this particular question? So the genetic setup, the gene setup for PBR is like this. If this is the PBR322 and this is ampicillin resistant gene, this is tetracycline resistant gene, this is ORI, this is ROPE. The genetic, research insertion site combination will be like this: PVU1, PST1. Within the ROP gene, you have PVU2. Within TET R, you have SAL1, BAMH1, and in between AMPAR and TETR, ampicillin resistant gene and tetracycline resistant gene, we have HIND3, CLAV1, EcoR1. Okay. So if you are forgetting the chronology of the genes, you can remember in this manner. You are eating something. You are eating evening snacks, right? So you will start with from this particular segment. So you started your snack with pav bhaji, PVU pav bhaji. You know that dish. Then after one pav bhaji, you have eaten one plate of pasta. Pav bhaji pasta. Now after eating one plate of pasta, you need another pav bhaji. So PVU2 pav bhaji pasta pav bhaji. And after that, you need to eat some salad. Okay. So pav bhaji pasta pav bhaji salad. Then bam is bana ke. Then in Hindi class, you are studying economics. So in this manner, you can recall the chronology of the recognition sites of this particular plasmid PBR322. So that's why E. coli having PstI site is present inside the ampicillin resistant gene. So it cannot grow in tetracycline medium. So let me check. Okay, all of you have given answer. Option B. Assertion is false, reason is true. The recognition site for PST is present in the ampicillin region. That is true and that is false. Yes, all of you are correct for this particular question. Let's move forward. Question number 19. Yes. Now, the components of bioreactors. Question from bioreactor, another very important topic. Question is expected. I think must be you will be getting one question from bioreactor topic for this year. It has an aggregator system. Yes, agitator system means for shaking, and oxygen delivery system, foam control system, froth breaker or foam breaker, temperature control system, pH control system, sampling points of withdraw cultures periodically if you are performing continuous culture. So the answer is easier one. That's why I'm not sharing any poll for this particular question. So all are correct. All of these structures are present or components of bioreactor. Let's move forward to the next question. Okay. Now let uh, check this particular statements. It's about first statement is about restriction endonuclease function by inspecting the DNA and binding to it when they find their specific recognition sequence. Second statement is saying DNA fragments are positively charged molecule. Oh my god. DNA fragments move towards the anode under the electric field, and option 4 is saying the recognition site for restriction enzymes are palindromic in nature. Now for this question I am sharing the poll. You have to answer. Okay. Already Ayush, Dr. Grisma has given answer as option D. Let me check. Uh, let me wait for others as well. Okay. So restriction endonuclease function by inspecting, we have to search for the incorrect statements. So DNA fragments are not positively charged, negatively charged due to the presence of the phosphate group. DNA fragments move towards a node. Okay. So this is also false. Two, this is right. The recognition sites for restriction enzymes are palindromic. Okay. So this is also true, this is false. So 2 and 3, sorry 1, we have to search for, what was the question? Incorrect. Okay, which of the above statements are incorrect? The restriction endonuclease function by inspecting the DNA, binding to it when they find their specific recognition site. This is correct. DNA fragments are positively charged molecule. This is incorrect. DNA fragments move towards the anode under an electric field. This is correct. The recognition sites, this is also correct. So option B is our correct answer. Okay. I don't know, many of you have given D. Ayush has given as option B. That is correct. Coming to the next question, read the following statements. Now the question is saying restriction enzymes belong to a large class of enzymes called nucleases. Option 2 is saying hind 2 cuts DNA at a specific sequence of 6 base pairs. DNA molecules are hydrophilic in nature. Restriction enzymes cut the sugar phosphate backbone. Now, which of the above given statement are correct for the restriction enzymes? So, here we have to find the correct answer. Restriction enzymes belong to a large class of enzyme called nuclease. That's correct. Hind 2 cuts DNA at specific sequence of 6 base pair, and the base pair can be GTTAAC, right, for hind 2, and it will produce a blunt cut. DNA molecules are hydrophilic. That's also true. Restriction enzymes cut the sugar phosphate backbone of DNA. Okay, so that's also true. So all are correct. 1, 2, 3, 4. Option B. And none of you have given the right answer. Okay. Coming to the next question. Question 22. Which of the following vectors or techniques is or are used for cloning in eukaryotic organism? Okay. Gene gun, microinjection, TI plasmid, disarmed retrovirus. We have to search, we have to check for this particular point. You have to transform eukaryotic organisms, not prokaryotic. So you can transform eukaryotic organisms like plants with gene gun, animals with microinjection, TI plasmid agrobacterium based transformation to plants. Disarmed retroviruses are used for animals, to transform animals. So all of these methods A, B, C and D, all of these methods can be used to transform eukaryotic organisms, and 50% those who have answered, they were right. Question number 23. Okay, in genetic engineering, the antibiotics are used as sequence from where replication starts to keep the culture free from infection, as selectable marker to select healthy vector. Sharing the poll here. Now it's a very easy question. Antibiotic resistant antibiotics are used as sequence from where replication start. We call that origins or site, to keep the culture free of infection. For that purpose, already the culture chamber is sterile, culture medium is sterile. Okay, sterilized medium is used. So option B cannot be the answer. As selectable marker, as reporter you can use it. Okay, we have to check for the reporter genes or the marker genes like AMPAR, TET are antibiotics are not used to select healthy vectors. So C is our answer, and none of you are correct in case of this particular question. Okay, antibiotics are used to check either presence of antibiotic resistant gene is there inside the cultured bacterial colony or not. So antibiotics are searching for selectable markers. That's why yes, option C will be the answer. Coming to the next question, the colonies of recombinant bacteria appear white in contrast to blue colonies for non-recombinant bacteria. So colonies of recombinant will appear white and the non-recombinant will appear blue because of which of the following reason? Already we had discussed some similar sort of question, similar kind of question. I think you can answer easily. The method is called, the mechanism is called as insertional inactivation, right? It's about that lag-Z gene and the beta-galactosidase and the interaction with the chromogenic substrate. Okay, so option D is saying insertional inactivation of beta-galactosidase in recombinant bacteria. Absolutely. So most of you have given the right answer. Coming to the next question, which of the following enzyme is not involved in recombinant DNA technology? Which of the following enzyme? Exonuclease can be used, endonuclease can be used, ligase can be used. Now you tell me catalase, you can use catalase or not. Okay, already I got the answer from Ayush. That is catalase. Catalase is not used in rDNA technology. Catalase is a normal enzyme produced inside our cells and that is responsible for splitting hydrogen peroxide molecules. So here the answer is option D. Catalase is the enzyme which is not used for our DNA technology purpose. Question 26. Which of the following features of genetic code does allow bacteria to produce human insulin called humalin by recombinant DNA technology? Is it genetic code is nearly universal for that, or the genetic code is specific for that reason, or the genetic code is unambiguous or not ambiguous, or the genetic code is redundant? For which of the following reason this which of the following statement or will be satisfying our question? I am sharing the poll here. Which of the following features of genetic code does allow bacteria to produce? Means you can take gene of interest from any organism and you can insert inside another host or alien organism, and the way our gene inside our body producing human insulin, similar manner the same gene inside the culture of bacteria E. coli, they are also producing human insulin. So which of the following criteria is satisfying that? Is possible? No. Genetics, if genetics code is specific, yes, genetic code is specific, right? A particular genetic code, a particular triplet codon can produce or can code for a particular amino acid, that's correct. But the specificity is not for a particular species, right? The answer will be genetic code is nearly universal. Genetic code is nearly universal, and that is the reason even the cellular machinery of a bacteria can produce the similar protein by reading the DNA setup which is obtained from human gene. Why it's possible? Because the genetic code is universal in nature. Okay. the similar, that same 20 or 22 amino acids are produced during the time of central dogma for prokaryotes as well as eukaryotes. Next question. Polyethylene glycol can be used for which of the following purpose? Okay, I'm sharing the poll here. Polyethylene glycol, can you use it for energy production? Can you use it for gene transfer? Can you use it for biodiesel production? Or is it used for seedless fruit production, for parthenocarpy? For which purpose you can use polyethylene glycol? Okay, so the answer is, along with divalent cation, polyethylene glycol can be used. Now you can give me a quick answer. This is the hint. Sometimes polyethylene glycol is used along with divalent cation. Now can you write down your answer into the comment box? Can you guess the right one from these four options? Where in biotechnology we are using divalent cations? Have you studied the topic making the bacterial cell competent just prior to insert the DNA molecule? So that is used for gene transfer without a vector. Option B will be the right answer for this particular question. Okay. Uh, another method also we use polyethylene glycol, that is for when we are making the somatic cloning, right? We are hybridizing protoplast. Okay. Next question. Following statement describe the characteristics of the enzyme restriction endonuclease. You have to identify the incorrect statements. Okay. Let us read out the statements. The enzyme cuts the sugar phosphate backbone at specific site on each strand. Okay, we have to find out the incorrect statements. So this is correct. The enzyme recognizes a specific palindromic nucleotide sequence in the DNA. That's also correct. The enzyme cuts DNA molecule at identified within the DNA. The enzyme cut DNA molecule at identified position within the DNA. That's also correct. The enzyme cut the H bond at specific site in between G and A on each strand. So here I am sharing the poll. Let me check either you can answer this. Okay, so already I got one answer. Yes, these enzymes, they are responsible for cutting sugar phosphate backbone. They are cutting the phosphoester bond. They are not responsible for cutting the hydrogen bond, because it's a kind of weaker interaction, right? So the answer will be option 4. That's incorrect. Now moving forward to next question, question number 29. And can you see this question? This capacity, the capacity to generate a whole plant from any single cell of the plant is called differentiation, somatic hybridization, totipotency, or micropropagation. What do you think? What will be the answer? If you have only one single cell of a plant, by utilizing plant tissue culture technique, from that single cell you can produce a whole plant body. So what do you call that? Okay, I got answer from Ayush. Totipotency. Yes, this is the answer. totipotency. From one single cell, when you can produce a full organism, that capability is called totipotency. Yes, Ritika also got the right answer. Yes. Now, can you please tell me, ranking wise, which is below totipotency? Is it pluripotency, multipotency, or monopotency? So, below totipotency, the capability of producing few kinds of tissue, that is called as pluripotency, few kinds of cells called as multipotency. We don't have totipotent cells present inside our body. We had few totipotent cells during the time of our early embryonic development, like our zygote is totipotent cell, or the initial blastomeres, 2 cell stage, 4 cell stage, 8 cell stage, those cells were totipotent or pluripotent in nature. Okay. Now let's come to question number 30. Which of the following techniques does not serve the purpose of early diagnosis of disease for its early treatment? Enzyme linked immunosorbent assay, recombinant DNA technology, serum and urine analysis, or PCR technique. One of the technique you cannot use for early diagnosis. Yes, we call those potent cells as stem cells. Okay. So this question is from biotechnology and its application chapter. So Ritika has got one answer, and the answer is serum and urine analysis. Yes. So serum and urine analysis is one of the conventional technique to diagnose disease, but it takes time. Now, for rapid diagnosis, you can use RDNA technology, you can use enzyme linked immunosorbent assay, you can use PCR. Like RT-PCR is one of the method which is used to detect that COVID, right? If you can remember those COVID days. Now, question number 31. We have to select incorrect statement regarding chemical structure of insulin. Okay. So, I am sharing the poll here. Mature insulin molecule consists of 3 polypeptide chains A, B, C. Insulin is synthesized as prohormone which contains extra stretch of C-peptide. C-peptide is not present in the mature insulin molecules. Polypeptide chains A and B are linked by disulfide bridge. So, very easy question to answer. Okay, come on. Quickly you can give the answer. It's very easy. Ritika already got the answer. Let me check how many of you apart from Ritika can give the answer. So according to Ritika, mature insulin molecule consists of 3 polypeptide A, B, C. That's wrong statement, and rest all are right, correct. Okay. Only Ritika is giving the right answer. And yes, obviously, that's the right answer, as because mature insulin, immature insulin, or pro-insulin or pro-hormone, that has three of the chains A, B, C. When our body is maturing it, after the process of maturation, the active insulin, that's having only B chain and A chain, and they are connected by disulfide bonds. C chain will be cleaved. Next question. So you have to match the following. I am sharing the poll here. Match the following. Gene therapy is very easy. Gene therapy allows correction of gene defect. RNA interference is cellular defense mechanism in eukaryotes. ELISA a diagnostic test for AIDS, not confirmatory test. Remember, gel electrophoresis, that is a separation technique for of DNA fragments according to their size. So the answer will be A for 4, B for 3, okay, C for 2. So option C is our right answer. And let me check how many of you are right. So in the meantime, let me check the leaderboard who are doing absolutely awesome. Okay, why it's not coming? Okay, no issue. Moving forward to the next question, and the question 33 says, in gene therapy of adenosine deaminase deficiency, the patient requires periodic, focus on this particular word, patient requires periodic infusions of genetically engineered lymphocytes because genetically engineered lymphocytes are not immortal cells. A retroviral vector is introduced into these lymphocytes. Gene isolated from marrow cells producing adenosine deaminase is introduced into the cells of embryonic stages. Okay, lymphocytes from the patient's blood are grown in culture outside the body. Now, why exactly there is need of frequent infusion of genetically modified lymphocytes in the body of the patient? Here we are not talking about the embryonic phase of the patient. Okay, remember the 4 years old child, the first instance of a genetic the gene therapy, ADA, which is a very crucial enzyme we need to produce, and that enzyme will be helping you for T cell maturation, right? Now, you know that how crucial and how important are that T cells are for our immune system. They're crucial part of your innate immunity, right? So I got the correct answer from Ayush, Ritika and Dr. Grishma. Yes, genetically engineered lymphocytes are not immortal cells. This is the correct answer. And Ayush and Ritika given the right answer. And as because we know that lymphocytes are nothing but WBC, and they have lifespan varying from 10 hours to 10 days, right? So after 10 days, these lymphocytes, 10 or 12 days, these lymphocytes will die inside the body. Now, again, you have to replace the new set of genetically engineered or genetically modified lymphocytes, and they will be producing this adenosine deaminase enzyme for certain period of time. Okay. Next, question 34. Which of the following are fused in somatic hybridization involving two varieties of plants, pollens, callus, somatic embryos or protoplast? Now this is easy question. Okay, I'm expecting correct answer for this particular question. So pollens, there will be no fusion of pollens, there will be no fusion of callus, there will be no fusion of somatic embryos, right? For somatic hybridization, you need fusion of protoplast, and the methods are used, that is chemical fusion method or electrofusion method. For chemical fusion method, we use polyethylene glycol, and for electrofusion, we use electric shocks. Okay, Dr. Grishma and Ritika again given the right answer. Protoplast are fused during the time of when you want to produce somatic hybridization technique, right? 35, question number 35. The laws and rules to prevent unauthorized exploitation of bioresources are termed as? Another easy question. Okay, so the laws of rules of prevent unauthorized exploitation of bioresource are termed as. Here we are talking about laws and rules. Okay, here you are not stealing something. If you are stealing someone's intellectual property right, then that will be considered as biopiracy. So all these terms will come under bioethics. Okay, and that's why this will be the answer, option A. Bioethics will be the answer. Biopiracy, when you are stealing something, stealing somebody else's work or somebody's invention. And biopatenting, okay, implementation of the rules or regulations comes under bioethics. That is called biopatenting in a legal manner. So for this question, answer will be option A, bioethics. Move forward to the next question. Given below are two statements, another statement based question. Statement 1 is saying 27 documented varieties of basmati are grown in India. Statement 2 is saying genetic modification has decreased the efficiency of mineral usage by plants. I'm sharing the poll here. I think it's a kind of easy one, but information based question. If you can remember the lines of NCERT, then only you can answer this. So, yes, there are 27 documented varieties of basmati that is grown in India. That is true statement. Genetic modification has decreased the efficiency of mineral usage by plants. No, that has increased the efficiency of mineral usage. So, statement 2 is false. So, that's why the answer will be statement 1 is correct, but statement 2 is incorrect. Option A. Yes. And two of you, Ayush and Ritika, got it correct. Moving forward to the next question. Another statement that is saying that cry1AC gene controls the cotton bollworm while cry1AB controls corn borer. Okay, again, memory based question. Statement 2. The process of RNA interference requires the introduction of only sense RNA. Let me check either you can answer these or not. So the cry1AC, if you remember the lines of NCERT, cry1AC and cry2AB, cry1AC and cry2AB, these two genes control cotton bollworm, while cry1AB controls corn borer. That is true. Whereas statement 2, the process of RNA interference requires the introduction of only sense RNA. No, it requires mainly the antisense RNA, and here we require double-stranded RNA, which is having only one sense and another complementary antisense RNA. So that's why the statement 2 will be false. So statement 1 is correct and statement 2 is incorrect. Okay. Moving forward to the next question. Question number 38. Given below are two statements. Statement 1 is saying the C peptide is crucial for the biological activity of mature insulin. And statement 2 is saying in India, 12 recombinant therapeutics are presently being marked. Again, I am sharing the poll here. Another memory based question from this particular chapter. Probability is there, mostly you will be getting memory based question from biotechnology and its application. Though few concepts are there, but mostly things are memory based. Biotechnology Principles and Processes, the first chapter, that is conceptual chapter. The second one, the application, that is memory based chapter. Okay. So for these particular questions, C peptide is crucial for the biological activity of mature insulin. That is absolutely false. Whereas in India, 12 recombinant therapeutics are presently being marked. That is true. So statement 1 is incorrect. Option B will be the right one. And let me check how many of you marked it correct. All of you have marked correctly. Coming to the question number 39, assertion and reason based question. And the assertion is saying RNA interference involves silencing of a specific mRNA due to complementary double-stranded RNA molecule that binds to and prevents translation of mRNA called silencing. Reason is saying RNAi takes place in all prokaryotic organisms as a method of cellular defense. Okay, now that's easier one. Let's solve this one. RNA involves silencing of specific mRNA by utilizing double-stranded RNA molecule. Okay, so assertion is true statement. RNAi takes place in all prokaryotic organisms as a method of cellular defense. Reason is false. RNAi, there is a method that is adapted by eukaryotic organisms. So that's why assertion is true, but reason will be false. Option A, yes. So all of you have given right answer for this particular question. Now question number 40, again another assertion and reason. The Indian Parliament has cleared the second amendment of Indian Patent Bill. And reason is saying it takes many issues into consideration, including patent term, emergency provisions and research and development initiative. The poll is here for your answer. Ayush is saying C. Okay. So let me check. Assertion is absolutely true. Reason, that's also true. And the Indian Parliament has cleared the second amendment of Indian Patent Bill because it takes many issues into the consideration. Yes. Option C is our correct one. Moving forward to the next question. The question is here, another assertion and reason. We can obtain diseased plant from a virus-free meristem. The reason is saying the meristem, apical or axillary of a plant infected with a virus is free of that particular virus. Again, I am sharing poll for this particular question. So if we check the assertion here, we can obtain disease-free plant from virus-free meristem. Something is missing. Something is wrong in this assertion. What is missing? We cannot obtain diseased plant from a virus-free meristem. Rather, we can obtain a virus-free plant from a diseased virus-free meristem from a diseased plant. Yes, so that's why assertion will be the false. From diseased plant you can obtain diseased plant. So if you have to replace this particular words, we can obtain virus-free meristem from diseased plant. And the meristem of plant infected with the virus is free of that particular. So this is true. So assertion is false and reason is true. Option B. Let me check how many have marked it correct. All of you have marked as correct. Next, assertion and reason. It's saying genetic modification of crops will enhance the nutritional value of food. Golden rice is genetically modified crop enriched with vitamin A and iron. I think this is an easy question. You can answer it very quickly. Genetic modification of crop will enhance the nutritional value of food. That's a true fact. And the reason is saying, yes, one of the example is golden rice. We can justify it that golden rice is genetically modified crop which is enriched with vitamin A and iron. That's also true. So both assertion and reason are true, and the reason is the correct explanation. Okay, so Ayush was really quick while giving the answer. Coming to the next assertion reason, another assertion and reason, and the assertion is saying the milk of first transgenic cow Rosie is better than natural cow milk for human babies, and the reason is saying it produces human protein alpha lactalbumin enriched milk. Okay, if you have studied the NCERT in proper way, you can really answer this question. The milk of first transgenic cow Rosie is better than natural cow milk. Why? Because it produces human protein alpha lactalbumin. So for this question also C will be the correct answer, and I think I got the answer from Ritika. Yes. Now moving forward to next assertion and reason, and the assertion is telling us early detection of disease using conventional methods is often not possible. The reason, conventional methods rely on visible symptoms which occur only when pathogen concentration are high. Direct lines from NCERT. So try if you can solve this particular assertion and reason, though it's kind of easier. I'm getting mixed kind of answers. Someone is answering A, someone is answering C. So early detection of diseases by analyzing urine or saliva or blood, conventional methods is often not possible. That's absolutely true. Why? Because conventional methods rely on visible symptoms which occur only when pathogen concentration are high. That's why we'll be taking help of uh, recombinant DNA technology, PCR, ELISA. So both assertion and reason are true, and the reason is correct explanation. Yes. So here I got right answer from Ritika, uh, Mrityunjaya, and Dr. Grishima. Next question, moving to the next one. Now again you have to read out the statement and you have to find out the incorrect ones. So the question is saying the genetic engineering approval committee evaluates the safety of introducing GM organism for public use. transgenic animals had their DNA manipulated to uh, possess and express an extra foreign gene. Transgenic animals are engineered to be less sensitive to toxic substance than non-transgenic animals. 95% of all existing transgenic animals are mice. In India, 21 recombinant therapeutics are presently being marketed. So here is the poll and it's time for your answer. You can hit the answer button in the comment box. Okay, so the Genetic Engineering Approval Committee evaluates the safety of introduced GM, that is correct. Transgenic animals had their DNA manipulated, yes, that's also right. Transgenic animals are engineered to be less sensitive, no, more sensitive to the toxic substances than in compared to non-transgenic ones. 95% of transgenic animals are mice, that's also right. In India, 12 recombinant therapeutics are presently being marked. So that's why we got our answer as option A. And Ritika, again Ritika, Dr. Grishma and Mrityunjaya given the right answer. Question number 46. How many of the given techniques are used for early detection of disease in our body? Now this is kind of easy one. How many of these given techniques are used for early detection of disease in our body? If you don't want to go with conventional techniques. You cannot take urine, you cannot take saliva, you cannot take blood. Recombinant DNA technology, can it be possible? Serum analysis, urine analysis, PCR, ELISA. Okay, so you are answering as option C. RDNA technology can be used for as a tool for early detection. PCR, ELISA, absolutely. Option C. And again, Ayush and Ritika given the right answer for this question. Now coming to question number 47, again we have to read out these following statements and accordingly we have to find out the incorrect ones. So it's saying that most Bt toxins are not insect group specific. Flies and mosquitoes are diphterans. An explant can be used to generate a whole plant. Plants developed through micropropagation are genetically dissimilar to parent plants. Okay, so answer for this particular question, please write down your comments or answer into the comment section. Most Bt toxins are not insect group specific. Is it true? No, this is false. Incorrect. Flies and mosquitoes are diphterans. Yes. An explant can be used to generate a whole plant, obviously due to its totipotency. Plant developed through micropropagation, one of the method of asexual reproduction. So they are similar, they are not dissimilar. So that's also incorrect. So option 1 and 4, option 1 and 4, they are incorrect one, and we got the right answer, that is option C. And 50%, let me check, Mrityunjaya and Raj has given it the answer as right. Coming to question number 48, Bacillus thuringiensis forms the protein crystals which contain a toxic insecticidal protein, and this protein is, we have to find out the correct statements. Starting the poll here and reading the statements. that is activated by the alkaline pH of the gut of the insect pest? Yes. Binds with the epithelial cells of the midgut of the insect pest and ultimately killing it by making pores? Yes. Does not kill the carrier bacterium which is itself resistant to this toxin? Okay. So here all are correct. So D will be right answer. D will be the right answer, and let me check. Ayush has given or marked it right. Question number 49. Okay, second last question. Transgenic animals are produced to study normal physiology and development, to study disease, yes, to obtain useful biological product as given in NCERT, to test the vaccine safety, to test the chemical safety. So 1, 2, 3, 4, 5, option D, that is the right answer for this question. Now coming to the last question for today's series. Again, statement is given. I'm sharing the poll here. So it's we need to talk about Rosie, the first transgenic cow. So its milk has protein content of 2.4 gram per liter. Its milk has human alpha lactoglobulin. Its milk is more balanced diet than normal milk. Its milk has excessive cholesterol. Okay, so I'm getting the answer as option D. So its milk has protein content, that is right. You have to find out the correct options. Its milk has human alpha lactoglobulin, that's wrong. Having lactalbumin. Its milk is more balanced diet, that is also right. So 1 and 3. 1 and 3 are right, and milk has excessive cholesterol, that's also false. And let me check how many of you have given the correct answer. Ritika, Dr. Grishma and Ayush. Okay, that's great. So for today, that was all for my side, and this is the leaderboard for today's session. So average number 1 is Ritika. Thank you very much for attending this session and participating actively. Ayush, similar. Thank you very much for active participation and rebrushing your uh, practicing speed. Then Dr. Grishma, Raj. Dr. Grishma also was continuously answering in a proper way. Raj, Pastel, Mrityunjaya, Shaurya, Christina, Nagamma, and Sangeeta. Okay, thank you everyone. Thank you very much. So after this session, I'll be sharing another 50 questions into our WhatsApp group, and that question along with the solution, obviously along with the solution, and that particular question paper practice sheets that will act as a homework for you, and you can practice. You can check inside our WhatsApp channel, right? If you are not there, please go there, check for WhatsApp channel link. Into this particular channel, you will be finding the WhatsApp channel link of PW NEET English, and after every lecture we are uploading 50 questions and another 50 questions, and the toughness level will be little bit harder, and more assertion and reason based questions I have tried to kept over there. Okay. Okay, then thank you very much. Good night. Bye bye.